Hi, this is podcast number 4, week 2, cell and molecular biology. And in week 1, we talked about just rules in the syllabus, and then we talked about COVID. And now it's more traditional how this course starts. But as we go along, as I see a topic and how it relates to the pandemic, I'm going to talk about that. So this is just a series of images to give you an idea of how diverse cells are and their shape. So we're going to use a lot, like a general diagram of an animal cell, a general diagram of a plant cell, a general diagram of a prokaryote, and they don't encompass the diversity of cell shapes and sizes that one sees in all organisms. So you can have long, thin cells, corkscrew shaped cells, cells that are shaped like lifesavers, flattened cells, cells that have specialized grabbing structures on one side, cells where the two cells that are interacting are vastly different in size, like an egg and a sperm. You have cells that have some kind of way of propelling the cell through a fluid medium, like Chlamydomonas. You have plant cells that have cell walls and can be very boxy-like and rigid. And you can have very spider-like anastomosing cells with all kinds of processes like neurons. And if you notice the scale bar here, 5 microns, 100 microns, 50 microns, so on, they're really different in size. So like this is 20 times bigger than that, for example. And there is huge diversity in cell size. On the one extreme end are really long cells, like in your nerve cells. If you're a short guy like me, they're not as long than if you're a tall person. But humans have a cell in their lower back where the cell body of the neuron is cell body with the nucleus and most of the cytoplasm, and then a very long process coming out of that cell body called an axon, and that will go all the way to a person's big toe. So in somebody like LeBron James, that could be, 3 or 4 feet, and it's all one cell. So if you think about, you typically don't think of cells being a meter long, but there are cells like that. And then this is often given as the example of the biggest extant cell, non-extinct cell, and that is an ostrich egg. But it's a little bit of a bait and switch here in that the germinal spot, which sits on top of the yolk in a giant egg like this, is really where you see the cell bits that you normally associate with cells. And then the yolk and the albumin, this is the white part of a fried egg, and this is the yellow part of a fried egg. They are technically part of the cell, although not everybody considers them part of the cell. So it's definitely a special case, as is this is in a special case. But this is less of a special case in that all the organelles that we're going to talk about are get shipped all the way down to your big toe from your cell body, and that doesn't happen in these giant eggs. But nonetheless, cells can be really big, but most of the cells we're talking about are pretty small. So again, we're going to use this generalized diagram of a cell over and over again, but I want you to remember that there's lots of cell diversity. But in a generalized diagram of a cell, 20 microns across is a typical cell size for an animal cell. A micron is 1/1,000,000th of a meter. I like to use a mitochondrion as a sort of yardstick. So there will be several mitochondria within a 20 micron cell. In a plant cell, that's going to have a rigid cell wall made out of carbohydrate. They tend to be a little bit bigger, maybe 20 by 30 microns in this rectangular sort of cell, and a lot of that is going to be made up of a fluid-filled vacuole in the middle. Nuclei tend to be lots bigger than mitochondria. Chloroplasts tend to be bigger than mitochondria. And mitochondria tend to be about the same size as a bacterium or as a prokaryotic cell. So it's important to know, the relative size of things as we're talking about these things. And as you study these, you get more of a sense for how big is this thing relative to other things. So those giant cells that we talked about on this previous slide, way, way, way bigger than is typical. And then within a cell, you also have structures that can be resolved by microscopes, electron microscopes, light microscopes. And it's good to have an idea of about how big are we talking about here. So ribosome is actually a pretty big thing. This is a protein and nucleic acid structure that is responsible for translating messenger RNA into protein. Every cell has thousands and thousands of ribosomes in it, made up of a large subunit and a small subunit, and they're about 25 to 30 nanometers across. So nanometers is 1,000 times less than a micrometer. Okay, so this is 20 microns. This would be 20,000 nanometers. So nanometers are smaller than micrometers. Cell membrane is about 7 or 8 nanometers thick. A microtubule is considered a large diameter filament. They're about 25 nanometers across. A microfilament is considered a thin filament. This is actin polymerized into the filament, and that's about 7 nanometers across. And then a DNA helix, when it's unwound, is about 2 nanometers across. And if you look at a scale here, that is a log scale, so each division on the scale is a 10-fold jump in size. This is 10 times smaller, 10 times, 10 times bigger, 10 times bigger, and so on, all the way up from here to here. On this end of the scale, we're talking about atoms. On this side end of the scale, we're talking about the scale of an individual human. Most of the cells that we're going to talk about are in this area of this diagram, and prokaryotic cells are 10 to 100 times smaller than that on average. There's examples that are that break this rule, and then viruses 10 times smaller than that. And again, there are examples of that break that rule too. So obviously we are under the scourge of the coronavirus now. Part of what makes viruses hard to work with is that they're really small. So techniques that we would use to, say, concentrate a whole bunch so that we can work on them are more complicated when you're talking about a virus versus, say, mitochondria. So if you use a centrifugal field and a centrifuge to pellet mitochondria so that you can work on mitochondria, you can also use a centrifuge to pellet large concentrations of virus, but it takes a lot longer and a lot more force just because they're lots smaller. Okay, the techniques that we'll run into in terms of viewing cells are many different kinds of microscopy. A bright field microscope is like those old microscopes that you would get in principal's lab, or in Cell Phys Lab, where you're just using white light to illuminate the specimen, and it hasn't been stained at all. And you can usually make out structure, but it can be hard to make up individual fields, individual organelles, that sort of thing in the structure. If you add stains, these are chemicals that are attracted to certain chemical properties, such as um, a positive or a negative charge. And they will add contrast to your sample, so you tend to see things better. You can do some manipulation of the light that is going through the microscope and increase contrast that way. That's called phase contrast. Another way of doing that is called DIC, or differential interference contrast. You can get more of a 3D sort of shape when you're looking at these things. A very common technique is using fluorescence. Fluorescent molecules are very, very sensitive in that it does not take much of the molecule in order to result in a very bright signal, and they come in color, so it can be easier to distinguish one thing right next to another thing if one thing is, say, yellow and the thing right next to it is red. And then a relatively new technique compared to the others, although it's been around for, I don't know, 30, 40 years, is called confocal microscope. And one limit in your ability to see things at very high resolution, be able to tell the difference between one line and another line versus the two lines blurring together, your sample has to be really thin so that light can pass through it or electrons can pass through it if you're using an electron microscope. One way to get around that is using a confocal microscope, where you use a laser to excite just one thin layer in a thick sample. So you don't have to physically slice the sample, which can be technically difficult, but instead you use a laser just to illuminate just a couple molecules worth of thickness in a sample. And then you can move which layer you illuminate as you move up and down and focus through a sample, and then use a computer to stack all those images back on top of each other, and you get like a 3D sort of picture. And confocal is the way to go for most cell biologists, in that they have to have access to a confocal microscope, but they're expensive. So I have dollar signs here, sort of like we were going to a restaurant. This would be Swenson's, and this might be Ken Stewart's. So a bright field microscope, a decent one, $500 or $600 a piece, depends on what you consider decent. That's about what they would be in an undergrad lab, for example. Phase contrast, you're talking in the low thousands of dollars. Same with fluorescence microscope, 10,000 or around there. But confocal, you're talking about 250 to 500,000, depending on its capabilities. So lots more money. A common technique that we'll see a lot is immunocytochemistry, immunohistochemistry, fluorescence microscopy, where an antibody, which is a protein molecule, it actually is shaped like a Y. They're typically drawn like these Ys. And the part where they bind or whatever they're going to bind to is at the tips of the Ys. And then on this part here, you can add a label of your choice, and fluorescent labels are often used. So antibodies are made by your B cells as part of your immune system. B because they mature in the blood, and they can be very specific. So this part of the molecule can rearrange its shape to literally bind to almost any shape, if not any shape, that we know that can exist. So they can, in the processing and the development of antibodies, the DNA actually rearranges itself to come up with different shapes, and that's why there can be things that have never existed on Earth before, and yet your immune system can make antibodies against it. Because antibodies can be very specific, they can be great as labels. And so if you want to know about a protein that is only expressed in the inner mitochondrial membrane, you can develop an antibody that will tag that protein and not see anything else in the cell, and then put a big fluorescent marker on it and come up with pretty pictures as a result of that. So antibodies are used a lot as a technology to image cells. Here's an example of different antibodies using on the same cell with different colors. So these green antibodies here are this green color here are labeling the protein tubulin and labeling microtubules, and then the red is labeling actin, and then yellow is labeling some kind of chromosomal protein, probably a histone. And in one image, this would be a confocal image where you take different layers of the cell, excite different layers of the cell with a laser, and then you reassemble the image after the fact. You can see a lot of different things inside of a cell all at once. This is the nucleus inside a cell, and then these cells are dividing. Okay. Transmission electron microscopy does not use light. Instead, it uses a beam of electrons that are shown through a cell. Transmission electron microscopy needs very thin sections, slices of cells. These slices of cells are typically only a few molecules thick. It is technically tricky to generate these slices of cells. Use a literal diamond knife. So the knives that cut these very thin sections are very expensive because they have a diamond coating on the edge of them in order to get very, very thin slices. And those thin slices are needed so the electrons can get through. The samples are stained with a stain that is electron dense to provide contrast. So like membranes are stained dark, and cytoplasm is stained not as dark. Proteins are also stained dark. And then you can see things at a smaller resolution than you can with the other technologies that I talked about that depend on visible and fluorescent light. Here is an example of a cross-section through a mitochondrion, where you can see the outer membrane of the mitochondrion and the inner membrane cristae that is all folded up, and then you can actually see mitochondrial ribosomes in there too. There's also scanning electron microscopy that looks at the surface of things. These are pretty spectacular sorts of images. You've seen pictures of bugs and eyes and that sort of thing using scanning electron microscopy. The way these work is whatever you want to see in 3D is first coated with a conductive metal. Typically it's gold. And the electron beam is focused on the outside surface of this thing, and then the electron conducting metal conducts the electron beam away from the sample, and then the detectors detect how the electrons bounce off that, and you can construct a 3D image. And then in a computer, you can add false color so that you can get these sort of things. So you can add false color on a yellow spectrum, so you see different contrast colors of yellow. And that's just to help you see it. It doesn't look like that on the screen while the sample is being taken or imaged. is post-processing sort of thing. But this sort of thing you see a lot. And the images that you've seen of coronavirus, if it's not a cartoon, if it's actual picture of the coronavirus, those were taken with transmission electron microscopy because it's a small thing. Okay, that's the end of the podcast